Hi class, so in the second video for chapter 2, it talks about what prokaryotes are. Prokaryotes are simple unicellular organisms. They only have one cell. They have no internal compartments. And that'll be more clear as we talk about prokaryotes and eukaryotes. The first assessment statement states that you have to draw and label a diagram of the ultrastructure of E. coli as an example of a prokaryote. There are many kinds of E. coli, but it's just a general name for a type of prokaryote. So we'll draw a picture here. Draw something that's like a little rod shaped, and this is the cell wall. Just inside the cell wall, you may be able to see a plasma membrane. Basically, it's like it has two little layers that are on the outside of the cell. There's usually some kind of little tail on a prokaryote, and that's called a flagella. And those have different functions than if it has little hairs that are coming off the side. Those are called pili. There is some DNA inside the cell, and that's found in the nucleoid region. And then there's some dots in here, and those are called the ribosomes. The second assessment statement states that you have to annotate the diagram from 221 with the functions of each named structure. So, first we have the cell wall. It is the barrier to the outside of the cell, and it maintains the shape of the cell. The plasma membrane regulates movement of materials into and out of the cell. The flagella is involved in movement of the prokaryote, and the pili is involved in transfer of molecules from one prokaryote to another. In fact, prokaryotes could undergo sexual reproduction through the use of these pili. The nucleoid region contains DNA, and this DNA has no proteins associated with it. And this is different than the eukaryotes, which you can learn in the next video, have proteins associated with their DNA. This DNA is considered free-floating. In the cytoplasm, which again, the cytoplasm is this liquid-filled area inside the plasma membrane. The ribosomes are the protein factories inside of a cell, so they produce proteins. The next assessment statement states that you have to identify the structures from 221 in electron micrographs of E. coli. An electron micrograph is basically a name for a picture that's taken by an electron microscope. which has a very high magnification. So you can see that on the outside is the cell wall, and the plasma membrane is going to be right up against the cell wall, but just inside of it. And you can kind of see this little squiggly line here, and that's the plasma membrane. It's labeled here. The cytoplasm is this fluid-filled space inside the plasma membrane. The nucleoid region contains DNA that doesn't have any proteins associated with it. So in this picture it's called naked DNA. So it has no proteins bound to it. And then there's a lot of these little dots. There's going to be hundreds of them. And those are ribosomes, and ribosomes make proteins. In the last assessment statement for this section, it says, state that prokaryotic cells divide by binary fission. And binary fission is a process where the cell duplicates its DNA, copies it in two, and then each resulting cell gets a copy of its DNA, and then it cuts itself in half after it grows. So you have two cells that are smaller than the original cell, but they'll eventually grow and divide again. So this is a nice picture here. You can see three different cells that are undergoing binary fission. You can also see the flagella here, which is involved in the movement of the prokaryotes. This is the second video for chapter 2. Please keep reading and watch the remainder of the videos. Thanks.